this YouTube video that was apparently taken yesterday did not really give my voice, so I tried now with this microphone, and today we will go away from phosphorus and phosphate, and we will have a look at the second, I would say, other most important macronutrient that is important in agriculture, which is nitrogen, which probably globally is the most important of all. So, does it work? No. Sé que el portero no funciona. Start with the slide to recapitulate what we have already seen on Monday. We have here our well-equipped farmer fertilizing the field. And this is a composite fertilizer with many nutrients, NPK typically. And we discussed already that the problems for phosphorus are mostly localized in the soil, in absorption of the phosphate to soil particles, and the bad bioavailability of this phosphate. Nitrate, the situation is a little different. Phosphorus as an element in the form of the nutrient phosphate is very immobile in the soil, sticks on the surface, is very mobile in the plant, is not reduced in the plant. Nitrogen is different. Different forms of nitrogen, ammonium and nitrate, have different mobilities in soil, have also slightly different mobilities in plants, but both nutrients, nitrate and ammonium, are mobile in the plant. And the problems with the nitrogen is more that we have conversions in the soil between oxidized and reduced forms. We have reduction in the plant, but not oxidation, as in the soil, by the plant. Reduction of nitrate to ammonium. And, well, overall, I want to show you a little bit that the overall problems with nitrogen are a little different. I just want to make an example how much nitrogen deposits you typically find in a field that has quite some moderate concentrations of organic carbon. And I just have here such an example calculation for the nitrogen quantity in the soil. So for 1 hectare of a field, when you only look at the 10 uppermost centimeters, you may find quite a high organic carbon concentration, like for example 1.2%. And this must be, if you want to quantify the weight and the nitrogen later, you have to multiply it by density, the soil density, and then you come to something like 16.8 tons per hectare organic carbon. And typically you have in stable deposits of organic carbon in the soil, a ratio of carbon to nitrogen of 11 to 1 about. So in the end you end up with 1,500 kilograms per hectare nitrogen, which is quite a lot, and which, when we say an annual plant maybe takes up 150 kilos of nitrogen, then this should be sufficient for 15 harvests. However, not all of this nitrogen is available. It gets available during the season by mineralization, to ammonium, to nitrate, by microorganisms. And all this later has to be considered when the farmer wants to quantify how much nitrogen he has to fertilize, he has to quantify how much is already in the soil and how much will the plant need and how much do I have to supply with fertilizer. So in general, I just want to start with a very classical experiment from Rothamsted in southern England, where people did work on the major nutrients for farmers, NPK, nitrogen, phosphorus, and potassium. And what they basically got out in long field, repeated field trials, and what many researchers found out later and repeated again and again, is that crop growth in general, or plant growth in general, is optimal when all nutrients are supplied at optimal level. Well, that's not so surprising. The data that I want to show here is just some data of this Rothamsted experiment. Without phosphorus fertilization and without potassium fertilization here, you get more or less, well, the same amount of yield. Either you fertilize with ammonium or not, and you get even less when you fertilize with nitrate. When you supply phosphorus in this field, so this field was already depleted by several rounds of growth for these nutrients, and when you now supply phosphorus and ammonium or nitrate, then you get quite some yield, but you still are missing potassium. And the best yield you get apparently when you supply nitrogen, either in form of ammonium or in form of nitrate. And in this case, the yield was better with ammonium. This reminds us a little bit of our data yesterday. We also had these beneficial effects of ammonium with bioeffectors on the phosphorus uptake and growth in low peak conditions. So the message of this picture of this table is basically that plant growth is optimal when all nutrients are supplied at optimal level. So of course now it's difficult to really decide what is the real optimal level and how can we adjust fertilization in a way that the plants really have the optimal level. And therefore we have to know that when you look at the yield and the fertilizer application, you first get an increase of yield. This increase of yield, the more you fertilize, this increase of yield gets smaller up to a maximum, and beyond that maximum, which is typically in a range of 200 kilograms of nitrogen per hectare, goes down again and yield decreases again. So this law of the optimum is very, very long known in plant nutrition, but still it's problematic to really identify where exactly these concentrations for optimal growth are. So for the typical crop in Germany, winter wheat, which we sow end of September or beginning of October, we have quite some nitrogen uptake, of up to 200 kilograms of nitrogen per hectare per year over the season. And most of the uptake does not occur in winter, after we've sown the winter wheat. Most of the uptake occurs much later, when the growth is for some time exponential in spring, in March, April, May, or June even. And for much of the early growth, when we still have relatively small plants, the nitrogen of the soil reserves is sufficient. And later, with increasing soil temperatures, with increasing environmental temperatures, microorganisms get more active in the soil, and they also mineralize nitrogen from deposits, and we get more nitrogen from the soil stores. And only this red part here we have to supply externally with a fertilizer. And what we normally have as recommendations for the farmers is that we have one fertilization here in the very beginning here as a starter, then one fertilization in the maximum growth phase, and then one final third application just shortly before harvest when grain filling is maximum. And we call this a split application of nitrogen that is recommended to the farmers, and I come back later to this scheme again because probably it requires some adjustments to current conditions. Okay, so the yield that we can get with wheat typically is something like 8 to 10 tons. That's a reasonable yield in well-developed systems in general. Okay. What the farmers can do, they can use very fancy equipped material to measure the nitrogen status of their plant by optical sensors and so on. What we use in the laboratory is something relatively old, already available since dozens of years. It's the spud meter, and we can easily, by looking at the chlorophyll fluorescence, which correlates very nicely to the nitrogen status, we can easily measure the nitrogen status of the plant. Already visibly, we can see whether the plants are a little bit darker. They are probably better supplied with nitrogen compared to chlorotic plants, which are a little bit more yellow and more likely have a nitrogen deficiency. So these things are pretty cheap and also used by farmers. Of course, for a real cropping system, for a real farm, we need also some soil analysis, and we take the soils before the growth phase, sometimes in the growth phase. Depends a little bit on what we are interested in. We determine all these values here. What is important is this Ammonium method, where we measure the mineralization, more or less, of nitrogen, where we measure nitrate and ammonium in soil solution. So, we discussed in the last 2 days use efficiencies of phosphorus use efficiency, and I will have a very similar slide today with nitrogen use efficiency. But before we go there, I have a slightly different definition of nitrogen use efficiency here, and the global crop nitrogen use efficiency is here defined as the total grain nitrogen that is removed by the harvest minus the nitrogen coming from the soil. This we don't consider. Divided or compared to the fertilizer nitrogen that is applied. And when we calculate it by this equation, the current global crop nitrogen use efficiency is still below 50%, but it is close to 50%. And compared to previous decades, it has quite improved. It's still that half of the nitrogen is not used. That's still not very efficient. But we are about half of the nitrogen is used in this range. There was recently quite an interesting study that when you historically look at individual countries and the nitrogen use efficiency in individual countries, and you do this globally for all countries worldwide, what you typically see is something that in the beginning, these countries, when they're not very developed, they're quite nitrogen use efficient because they have not much fertilizer and don't use so much. Then they are use efficient. Then they go into a stage where they are wasting a lot and they are getting quite inefficient, and at higher developmental level of the country, typically the nitrogen use efficiency again is higher, it's better. But typically this levels up and we never get to nitrogen use efficiency of 100%. With very, very optimal conditions we can get up to something like 70%, but that seems to be the maximum level. The nitrogen use efficiency for cereals, most important crops worldwide, harvested on the largest area worldwide, was for decades in a range of 30, 33 or so %. This has slightly improved, but is still relatively close to that value. So cereals and wheat especially are still globally a problem with respect to nitrogen use efficiency. I would say that the situation of nitrogen differs in many ways from all other nutrients, because nitrogen nowadays is not a limiting resource. We discussed about phosphorus, that phosphorus, if we use it in the current quantities, will run out in about 250 years, according to the expectations of the stores and reserves that we have and the current use. For potassium, the situation is quite similar. It will also run out. There are some expectations it lasts a little bit longer than phosphorus. But nitrogen nowadays is an unlimited resource because we can synthesize nitrogen from air. And this situation that we have nowadays, since 60, 70 years in practice, and principally this Haber-Bosch process for the synthesis of nitrogen fertilizer from air, we have since about 100 years. This contrasts the situation that plants and crops were exposed for historical times during evolution. So during evolution, during most times of historical agricultural production, nitrogen always was limiting, was the limiting element for plants. Because we have gaseous losses, we have leaching losses, we have deposits, nitrogen was always a problem. And now it's not the case, unlimited. The problem with nitrogen is on a different limitation level, and that's this conversion of air nitrogen, N2, with the triple bond between the two nitrogen atoms. This triple bond is extremely hard to cut. So we need enormous amounts of energy. We need catalysts, high pressure, and this costs, well, estimates typically say something around 2% of global world energy. Global world energy, that's huge, yeah, the amount that is needed. We have these unavoidable losses, gaseous losses, nitrate leaching, deposits, nitrogen depletion. And this nitrogen over-fertilization, since about 60 years, altered ecosystems already, is still altering ecosystems, drives species extinction because many species, of course, as I just said, are evolutionary adapted to low nitrogen environments. If we now pollute the nitrogen everywhere, and everywhere is a high nitrogen environment, all these environment adaptations for 10,000s, 100,000s of years is lost and not useful because we have so much nitrogen. So we have a lot of species extinction, and surprisingly, nitrate or nitrogen has relatively little effect on humans. There are always some... Sorry? Direct effects, yeah. There are always some cases told that, well, in Europe or in Germany in the 50s or so, there were some blue baby disease associated with nitrate in drinking water, but this was actually some microbial contamination that was related to this. So there's not really a lot of toxic effects of nitrate. We don't exactly know how problematic nitrate to humans is. We use nitric oxide as a relaxant, signaling relaxant in our muscles. So our body even produces nitrate. And a little bit of nitrate is even beneficial to humans. So for human health, it's not so much a problem, but for the environment it is. A nitrogen fertilizer when you can calculate, or when you calculate, especially with the energetic cost that it has to produce nitrogen fertilizer, it's one of the major, it's the major variable cost to the farmer. And I would say it's still too cheap because all the environmental problems, secondary effects, are not included in the price, as always. So, where are the problems with too much nitrogen use? As typically in China, in India, there are quite some problems. The drivers are not so often really the farmers that we apply too much, and sometimes not very well-trained farmers. Yeah? This is definitely also in China and in India the problem. Insufficient training of the farmers. But overall global drivers are our meat-based diets and animal farming, because we need so much protein for as a feed for our animals, and this requires quite high amounts of nitrogen. But there are many regions in the world, as you can see, where there's not so much pollution with nitrogen and even nitrogen deficiencies. And the global 50-year trend of nitrogen application here up to about 2010 for maize, rice and wheat, oh, it's hard to see actually, you can see it's still somewhat rising, this fertilizer application per field. I would say, as with phosphorus, we use excessive nitrogen worldwide, especially because we have to produce the fertilizer from nitrogen from air, and this is energetically costly, and always the problem is we don't have enough energy and we just burn coal or we burn gases, and we do this excessively, as you know. I think we, again, for similar reason as with phosphorus and the limited resource phosphorus, we have to overthink our excessive lifestyle. In Germany, the situation for different fertilizers, nitrogen, phosphorus, potassium, and calcium, liming, is as drawn here from the 50s to about 2010. Again, it's a little bit older already, but it still shows the major features. In the 70s and the 80s, we had massive overuse of potassium and phosphorus. The farmers realized, the researchers realized, and the farmers realized, oh, their yields are not worse when they use less phosphorus and less potassium. And so since then, they use much less phosphorus fertilizer, much less potassium fertilizer. But for nitrogen, this has not much changed actually. For nitrogen, it stayed more or less high, this fertilization level. And also for liming, but that's a different story. So, how much nitrogen do our crops need? Well, it depends on the species. And it's typically something between 120 to up to 250 kilos per hectare. What the plants really need in season and what you really take off from the field with the harvest. And there are some plants that are extremely nitrogen deficient. And one plant that is not a grain crop for consumption, for eating, but it's a biomass plant, which in our tour here during the weekend with Victor, I saw this here also in Costa Rica a lot, miscanthus grass, a tall grass coming originally from China. And this grass is extremely nitrogen efficient, and many researchers have documented that, and I show you one example of this grass, which is a perennial grass. You plant rhizomes of this grass, and in the first year, the yield is not very high from this plantation, but in the second year, third, fourth year, you have almost optimal yield, which can be in the range of maize yields, maize biomass yields. It can be even higher, depending on the environment. And dry matter yield can be, here, let's say, up to 15 tons per hectare, which is quite substantial. And it's nice that you can use this biomass for example, for burning in ovens. When I saw this graph here on a conference in 2009, I immediately decided, yes, I have to work on this plant. This seems to be the solution for everything. If we understand that, the physiology behind, we'll understand how to use nitrogen more efficiently. What is so special about it? I want to try to explain what you see here. You'll see basically a field trial with 3 nitrogen levels of fertilization. The gray one is the 0 nitrogen plot. The light blue one is the 60 kilograms nitrogen per year per hectare. And this dark green is the 120 kilograms nitrogen per year per hectare. So what can you see after you started the plantation? This is a perennial plant. Yeah? You can harvest the biomass and the stems for 15 or for 20, sometimes 25 years, without much decrease in yield. You have to supply, in the meantime, somewhere a little bit of potassium and a little bit of phosphorus. But what you see is that both in the very beginning, the nitrogen fertilization level did not influence on the annual yield. This was something to me very surprising. And the other thing was the nitrogen from the field somehow was not depleted. And even at the end of these experiments, the 0 fertilization also gave the same yield as the high fertilization. So the question is, what is happening here? What is happening here in the end? Why is nitrogen not depleted? And here, why is this establishment without nitrogen so good? For this beginning here, there's a secret in this paper that I in the beginning did not realize. This field where the experiments were done was a field where for several years legumes were, which produced a lot of nitrogen, and the nitrogen load of this field was pretty high. So the beginning is maybe not so important. But why is nitrogen not depleted from the field? The reason is that we have roots, the rhizome structure, the stem-like structure, which grows below ground and which can store nutrients and remobilize nutrients. This rhizome allows nitrogen to be mobilized to the shoots in spring and summer, up to almost 400 kilograms per hectare per year are mobilized from this rhizome and then taken up from the soils surrounding here by the roots. And now comes the interesting thing. Now comes the fall. The leaves get senescent. The shoot stops to do photosynthesis. Mineral nutrients, especially the mobile ones, are mobilized to the rhizome, a few to the root. Those elements that are not very mobile, like micronutrients, iron, zinc, copper, these stay in the leaves. But the nitrogen is mostly remobilized. Some nitrogen stays also in this straw here, in the leaves. But after winter, because in winter it's also raining sometimes, nitrogen and micronutrients are washed out of the leaves. They end up in the soil below, and they're taken up by the roots again. Part is in the rhizome. The losses are extremely small, and a lot of nitrogen can be remobilized later from the rhizome. So it's a certain harvest system where we harvest the straw only after winter, when it has very, very low nutrient concentrations. It's mostly carbon, oxygen and hydrogen that you find in the straw. The leaves have fallen down, it's mostly the stems that you harvest. It is this remobilization of nitrogen during growth and after growth into the rhizomes that is the secret of this very high nitrogen use efficiency. But, as I said, when the plants are not yet established, this system cannot yet work. In the beginning, there must be some other mechanism, and one suggestion was that there may be some nitrogen-fixing bacteria that may contribute there. And we rarely find nitrogen-fixing bacteria associated with plants. We find quite some evidence nowadays, which are agronomically important in sugarcane. And sugarcane is a plant very closely related to miscanthus. Yeah? So there are some similarities. Okay, so we look at miscanthus in Hohenheim, and very similar to the field trials in southern England, we have field trials by some colleagues, for example, Lewandowski and some other colleagues. They maintain these field trials since more than, I think now it's more than 30 years. This is a field trial established since more than 14 years, where they have zero fertilization, intermediate and higher fertilization, and compare these year to year. And when you go there, the high nitrogen fertilization plots are next to the zero fertilization plots. And this is after 14 years of zero nitrogen fertilization, and you're surprised. It looks good. Yeah? They look perfect somehow. Yeah. So this is exactly a plot that we are interested in, that we wanted to use for further analysis. What we did was we harvested basically the microbiome and did pyrosequencing of basically different compartments to see which bacteria are associated with different compartments. Different compartments are the rhizome, the root, the soil, and the rhizosphere soil. Yeah, 4 different compartments. And when you do so, you see that by analyzing all the sequences that you get and doing some principal component analysis, you see that for the nitrogen 0 level, you get some data. For soil, rhizosphere, root, rhizome, and for nitrogen 80 kilograms per hectare, you get soil, rhizosphere, root, and rhizome data. And when you do a principal component analysis of this, what you easily see is that the bacterial sequences, when analyzed by this PCA analysis, that these bacterial sequences are very similar, the distribution is very similar in the different compartments. So in soil, independent of nitrogen, the samples are very similar. In rhizosphere, the samples are very similar. In root, this is root. This is rhizome, and this is soil and rhizosphere. So that's how the data is. When we now look at the different phyla that we find in the different compartments in soil, rhizosphere, root, rhizome, without nitrogen, with nitrogen, without, with, and so on. What can we see? We see, well, there's a little bit shift here in the different phyla. Moderate changes with nitrogen. It's not so much. It's surprisingly little what happens. And we have seen this in other experiments also. With different species and different farming systems, the nitrogen seems not to affect so much the microbiome of plant-associated and soil-associated fractures. Most differences seem to be here in the rhizome. And this we analyzed a little bit more in detail, and for example looked at those phyla and families that are suggested to be nitrogen fixing or may contain nitrogen fixing bacteria in them. And when you look in soil rhizosphere root and rhizome here, yes, in the rhizome there are some potential nitrogen-fixing bacteria enriched, proteobacteria enriched. So this seems to be interesting and would suggest that maybe the rhizome is a compartment for nitrogen fixation. How this works, we have no idea, because this rhizome is a pretty rigid structure, and there's not much oxygen and nitrogen diffusion into this rhizome, but it seems this compartment might provide maybe a similar environment as a nodule in young legumes for support nitrogen fixation. The direct amount of nitrogen fixation must be relatively low. Our colleagues from the agronomy department tell us that there are some kind of 40 kilos of nitrogen that they cannot explain, that must come from something, maybe from fixation. Yeah. We have tried to measure this. It's technically a little bit difficult, and we were not successful yet to really show how much of nitrogen is fixed, or whether nitrogen at all is fixed. An estimate is some minor part of the nitrogen, maybe some 30, 40 kilograms may be fixed, and part maybe in the rhizome. So when we have here these schematic plants with roots and laterals and rhizome, soil and rhizosphere, rhizosphere in white, the nitrogen 0 and nitrogen 80 kilograms per hectare conditions. Well, we see under these conditions there are biological nitrogen fixes apparently more abundant here. This sounds interesting. By contrast, in the NAD plots, roots and rhizome, we find that some denitrifier families are more abundant than in the N0 plots. And that's something also related, maybe diseases or disease protection, that is very difficult to say from this kind of analysis, but there's also some indirect effect, maybe also on the immune system. Okay, so these are relatively recent data, but we work on Miscanthus since much longer, and when we started, we searched in the literature whether there are already some diazotrophic bacteria. Diazotrophic means nitrogen-fixing bacteria known to be associated with Miscanthus. And yes, when we searched in the literature, we found one that was isolated by Tony Hartmann's group in Munich, close to our laboratory. And we directly called him and asked him on a conference, can you send this bacterium to us? We want to investigate it more. And Tony Hartmann was also very interested in this bacterium because this bacterium called Herbacterium friesingense, this bacterium had some similarity to azospirillum. It was found in sugarcane. And sugarcane also, as I said, is suggested to have some associated nitrogen fixation, probably. And there was also a Herbacterium xerocanica found in sugarcane. And this bacterium that they isolated in Freising, therefore it's called Friesingense, close to Munich, this had a lot of similar properties as the Herbacterium found in sugarcane. So there were these analogies, and it sounded very interesting, and this was a very promising bacterium for this nitrogen fixation. And when we worked on this, we actually did not really find that this bacterium, when inoculated in the plants, was helping to acquire nitrogen, but the bacterium was somehow improving growth. It was increasing root growth. It initially transiently it did some defense signaling, which we analyzed by RNA-seq. And later we found that this bacterium also helps somehow the establishment of total miscanthus plants from stems. So this bacterium was for us very interesting because it was isolated from miscanthus. It was also isolated from other biomass plants. And we thought, well, this is the one. And now we go back to our data here. And all these microbiomes. And to our surprise, we almost did not find it. It was almost not there in Hohenheim. It was there, we found some sequences that were corresponding to Herbacterium, but it was definitely a minor bacterium in this microbiome, in all compartments. It was an endophyte, so it enters the plant root, and it can be found in the shoot. Yeah, and it promotes growth, at least in some artificial conditions, as here. But we then worked again in the field. We did a very small plot. Well, part of the plot is shown here. And we established miscanthus from stems, not from rhizomes, which is relatively costly. From stems, you can establish them more easy. And we knew from greenhouse experiments, yes, establishment is helped by Herbacterium a little bit. So when we looked in the second year or third year, there was no benefit from Herbacterium inoculation anymore, so the biomass was more or less the same. But we did some analysis of these different fractions of the root, rhizome, rhizosphere, biosoil, as we had done in the other plot. And looked at the microbiome. And we do that, we look at the family level. What do we see? After 2 years, or in the second year after this inoculation, what do you expect? How much Herbacterium is still there? Nothing. Well, this was more or less what we found. There's almost no Herbacterium. We again found a few sequences, but really negligible. Yeah? Not much. And in the rhizosphere, in the soil, basically no difference. Yeah? Well, this may be expected because, well, if the Herbacterium is vanished, then all the other microbes stay the same, and, well, they should be the same. But in the root and rhizome, maybe we have some difference. And indeed, in the rhizome, we found some difference. Again, in the rhizome, which is probably the compartment where microbes cannot easily go so easily in. And what are the differences here? The differences were in some species, well, in the endorhizosphere communities most affected, which means root and rhizome compartments were affected. And in the rhizome, we had these here. It's hard for me to pronounce these names of these bacteria, but these are opportunistic pathogens. Yeah? And these were suppressed by Herbacterium. That seems to be beneficial. And Pseudomycete and Xanthomycete, plant growth promoting rhizobacteria, were enhanced. Yeah? So probably there is some beneficial effect on disease or health related issues in the rhizome for supplying somehow these internalized endophytic bacteria to other parts of the plant. Okay, so to summarize this, lessons from the zero fertilization trials and nitrogen fertilization and Herbacterium inoculation trials. Inoculations may be beneficial, maybe for plant growth and disease for establishment, but later they're not much beneficial, mainly for disease suppression, but we did not visibly see disease in one or the other plot. These inoculations, although the inoculant is lost after a while, there are long-lasting effects in the microbiomes, especially of the rhizome. And I would say the rhizome is an interesting candidate for some nitrogen fixation, although we could not really show that directly yet. Okay. With this, I would suggest that we make our first break already, maybe a little bit earlier. Am I so fast today? And then we look on ammonium and nitrate comparison. Our ammonium uptake is organized in plants and how plants respond to the takeaway of ammonium. Okay. transport, and the transport of ammonium especially. And I don't want to show you too much of this molecular architecture of the nitrogen transporters. I'll show you an overview from Arabidopsis. And this Arabidopsis plant is the model plant of the plant physiologists and the plant geneticists for several reasons. One reason was that the genome was apparently pretty small. It's still one of the smallest genomes that we find in the plant kingdom. And another thing was that this plant grows pretty fast and can have a life cycle that you get seeds after, oh sorry, there's some feedbacks. I don't know how I can solve this. Do you know how I can solve this? Feedback problem? This should be out, okay. Then I stand here. Huh? Okay, so we try. One step. So Arabidopsis now, yeah, small genome, that was important. Has a fast life cycle that's important for rapid publications. What else? Relatively small. That's a big advantage. When you have limited greenhouse space and you want to grow hundreds or thousands of genotypes, it's always good to have a really small, tiny plant. Yeah, that's the major advantages. There are also disadvantages with Arabidopsis. One disadvantage about Arabidopsis is that it, well, one advantage others would say it's closely related to rapeseed, to a crop. But in reality, many of its properties are very distant, very distinct from crops. So this plant makes a lot of seeds that are extremely tiny. That is good. This plant also is transformable. Genetic transformers can be done. You can get mutants, that's good. Another disadvantage that we only learned later was especially the one genotype that was used later for the first draft genome of a plant, the Columbia type, Col0 of Arabidopsis genotype, found or selected, collected initially in Columbia. Originally these even come from Holland, which is now known from sequence comparisons. Anyhow, these plants are not really, with respect to nutrient uptake, very similar to crops. They are not very similar to crops. These plants here, when you grow them under relatively, well, high nitrogen conditions, they accumulate in their leaves something like 6–8% of nitrogen. This is very atypical. In crops, the maximum nitrogen concentrations can be 3–4%. Yeah? So for nitrogen, it's not a very good plant for investigating the nitrogen efficiency and so on. Then what also came out was that especially the genotype that was selected for most of the experiments later for providing mutants to the whole Arabidopsis community, the genotype Columbia 0 was, of all genotypes that were investigated in more detail, the nitrogen, the least nitrogen use efficient one. So there are many disadvantages with using this plant, but still, we learned quite a lot from this plant, and some data I want to share with you today. And some data that I want to share with you now is, yeah, where do we find ammonium transporters? Other transporters I don't have here on the slide. We have in plants typically two gene families of nitrate transporters, so-called high affinity and low affinity nitrate transporters. These gene families have been quite in detail investigated. For ammonium transporters, we know that ammonium is flowing into the plant also by more or less two systems, a high affinity system and a low affinity system. This low affinity system is still genetically not clear what it is. The high affinity system is encoded by AMT, ammonium transporter genes, AMTs. And, well, these AMTs were identified around 20 years ago. And at that time, well, we did not know whether we have several families or so. So the researchers that gave the name to the AMTs gave the name AMT, or Arabidopsis thaliana, abbreviation for Arabidopsis, AMT for ammonium transporter, family 1, and then member 1. The researchers at that time did not know whether there are more gene families that later could be the name given AMT2. And indeed, there came up a gene family a little bit less related than these AMT1s to each other. This is a little bit more distant, but it's still the typical ammonium transporter. This was then called AMT2 or AMT21. And of these AMT transporters, we have in Arabidopsis only one member. And still, of this member, we know relatively little. How it works is not so well established. Where it works is not so clear. In other plants, we have AMT transporters also, and they work in mycorrhiza, from where ammonium is transferred from the fungus towards the plant. There, AMT transporters help. AMT1 transporters, AMT11, 12, 13, and 15, in Arabidopsis, we have overall 6 ammonium transporters, AMT11, 2, 3, 4, 5, and AMT2, so in total 6 ammonium transporters. 5 of them are expressed in the root. And using Arabidopsis, we can relatively easily identify knockout genotypes for individual genes, so knockout mutants for AMT1. And when we investigate knockout mutants where this ammonium transporter is deleted, we find the plant grows normal. But when we analyze in detail, we find it takes up about 30% less ammonium via the roots. So ammonium transporter 1 is responsible for about 30% of uptake via the roots. When we look at the mutant of AMT12, we find basically the same. Ammonium transporter 2 knockout, 30% less uptake of the plant, but the plants look normal. When we knock out 1,3, again, plants look normal, again 30% less uptake via the roots. When we now knock out 1, 2, and 3 and AMT2 together, this does not seem to transport much. So 30% less, 60% less, 90% less. There are 10% less of uptake of ammonium. And what do we expect? How do the plants look? Normal. No phenotype. They take nitrate. Yeah, and that's enough for them. So, that was, for me as a researcher in ammonium, uptake a little bit disappointing. I was hoping a nice phenotype of these mutant plants. Yeah? But apparently, with ammonium, the plant can live without this ammonium transporter. Okay, we have ammonium transporters also in the shoots. Why? Because when we take up the ammonium into the roots, we have to transfer it later via the xylem to the shoots, and in the shoots we have to unload the ammonium from the vascular system towards the cells, uptake the ammonium, we have to reuptake ammonium from assimilated nitrate reduction and then reuptake of ammonium from this. And there these transporters participate in the shoot, but in the shoot there are only these three expressed, and one, 81,4, is only expressed in the pollen. The function there is also not clear. When you knock it out in the pollen is viable, no problem for plants. So basically you couldn't knock out all the ammonium transporters. You don't lose much. And nowadays we know there's also similar, or start to know this also for other plants, for rice for example. And the plants also look pretty normal. So not essential, these ammonium transporters. However, we still believe they are important. And some basic features of these ammonium transporters, which can be generalized to other species as well. We have typically one or two ammonium transporters. In Arabidopsis, it's two ammonium transporters, number 1,1 and 1,3, which look very similar and have a very similar expression in the root. And this expression is in the outermost cell layers, which is the rhizodermis, which is the cell layer that also contains the root hairs. And in these root hairs, the AMT1,1 and the AMT1,3 are mostly expressed, both at more or less equal levels. Here only AMT1,1 is shown. You can see AMT1,1 is shown here when you make a radial section of the root. It's here in these outer layers. It's also in the cortical layer. In Arabidopsis we have only one cortical layer, and then in the inner parts it's relatively little expressed. And the interesting thing about AMT1,1 and AMT1,3 is also that when you deprive plants from nitrogen, when you, for example, grow these plants in nutrient solutions and transfer these plants and their roots to a nutrient solution not having any nitrogen, then the gene expression, here shown by northern blot, gene expression starts to appear after 1 day a little, after 2 days a little bit more, after 3 days quite some substantial gene expression, and later it goes down. AMT1,2, this is the gene expression that is also found for AMT1,3, very similar. AMT1,2 responds a little bit different, not so much, maybe a little bit later, it gets also expressed. And the reason is that AMT1,2 is expressed in different types of cells, at least in the root tip. It is expressed in the endodermis and the cortex, where ammonium, after taken up by outer cell layers, is transferred further to the vascular system. Yeah, and across this Casparian strip, you have to have some ammonium transporter. This Casparian strip is impermeable to nutrients, and so this ammonium transporter is involved there. The affinity of these ammonium transporters, typically at the site most closely located to the soil, in the rhizodermis, the affinity is pretty high, 10, 50 micromolar km, which is the half maximal concentration. The concentration at which half maximal transport occurs, and this is the so-called Km. And the Km for this transporter is a little bit lower. Well, you have already, with high affinity, accumulated ammonium into the root, and then in inner parts of the root, you need not such a high affinity transporter, can be a little bit lower affinity, but with more capacity. So in shoots, AMT1 is found in several cells, AMT1,2 also in mesophyll cells, also in part in epidermal cells. AMT2,1 notably also is found at the end of the vascular strands, probably to take up and reuptake of the ammonium that is still found in the xylem cell. It has not been yet depleted from the xylem cell by other cells. Okay, so this architecture of ammonium transporters in the plant is quite similar in crops. However, every crop that has been looked at, maize, rice, many other crops, it always is slightly different. Yeah? So the numbering of these transporters is different. You typically have this distribution that you have transporters in outer layers and transporters in more inner layers. You have different nitrogen regulation by these transporters, but generally they are upregulated transcriptionally by nitrogen deficiency. But yeah, the real time course of this, depending on the crop, is quite different. And also the regulation of the resupply is often quite different. So when we now look at the proteins, we investigate the function of these proteins very often by heterologous expression of these transporters, either in yeast or in frog eggs and oocytes. And there we do electrophysiology and measure currents across these transporters. Ammonium currents, so ammonium is an ion, a cation, and this ammonium ion generates a positive current when it goes through the ammonium transporter into the cell. And this we can measure electrically, and I don't want to show you data here, but I just want to show you some summary of results, because I showed you that the ammonium transporters are expressed in certain regions in the plants, in certain cells. They have a certain transcriptional regulation, especially upregulation under deficiency, which makes sense, because if you have a nitrogen deficiency, you want to get more ammonium, and that's why the transcription goes up. But there's also a very important post-translational regulation. And this we have investigated, or are investigating since many years now, and we get more and more data. And not to confuse you too much, I just show you some summary slide of this regulation. And here you see the ammonium transporter number 11 sitting here, in this case, in the cortex cell membrane, where AMT11 and AMT12 are expressed, in some cortex cells at least. And these ammonium transporters are relatively complicated, interesting proteins. We don't have to go through all the details, but these are trimers. Trimers that are basically 3 subunits that form one transporter, one active transporter. And these can be identical subunits, or it can be AMT11 mixed with AMT12, giving a mixture of subunits, and each subunit has a pore for the ammonium ion to go to. So, and we have not only ammonium transporters in this cortical cell membrane, but we also have nitrate transporters. We have also water channels, which are not shown here. We have potassium channels there. And of course, the plant now needs not only nitrogen to take up nitrogen, but it has to balance the ammonium uptake and the nitrogen with other things. And what do we have to consider? Well, first, we have to consider the charge of the ammonium. The plant has to balance the uptake of positive ions with the uptake of negative ions. So when the plant takes up ammonium, it has to balance it somehow with nitrate. Yeah? And maybe it should shut off potassium channels that not too much positive charge flows in. So under nitrogen deficiency, the situation is in a way that the ammonium transporters are probably open. The potassium channel, well, is closed here, that not too much potassium flows in. A little bit potassium probably flows through this channel also, or to other channels. And some nitrate transport goes through the nitrate transporter, and this nitrate transporter has a very strange name, either called NRT1 for nitrate transporter 1, or a novel nomenclature gave it the name NPF6 semicolon 3. Okay, that's the number. Okay, so to balance the charge, ammonium and nitrate. So this may be the situation under low availability of nitrogen. So the plant has some high affinity uptake via the nitrate transporter and some high affinity ammonium uptake via AMPs. And if now too much of ammonium would be available, then the plant apparently likes to close the ammonium transporters. And these ammonium transporters are then shut off. And this is by a post-translational mechanism where phosphorylation of the ammonium transporter plays a role. Phosphorylation in the C-terminus, both in AMP1-1 and AMP1-2, they have the same site. This also applies for AMP1-3, but not for AMP1-5. And we have recently identified the kinase that is involved in phosphorylating this transporter and shutting it off. So phosphorylation switches off the transporter under higher ammonium availability because probably too much ammonium may be toxic to plants. Ammonium toxicity. But to maintain still some inflow of nitrogen, the nitrate transporter is switched into a mode that is a low affinity mode to allow a little bit of nitrate to flow in, and it opens the potassium channel that allows now potassium to flow in. And surprisingly, it's all done by the same kinase and by the same phosphorylation mechanism in different proteins. It's always the kinase CYP23. These CYP kinases belong to a family of kinases that are found in plants with about 24, 25 members. Yeah, and this 23 is one of the members of these kinases. These kinases come always as proteins that interact with the second subunit, the so-called calcineuron B-like subunit. Calcineuron B is also not a very nice name for this. Actually, it's proteins that you find also as a gene family, and in Arabidopsis there are also about a dozen of these CBLs. And this CIPK23 specifically interacts with CBL1 and or CBL9 for these potassium channel regulation and nitrate transporter regulation. But for ammonium transporter regulation, only one of these CBLs work, and it's CBL1. CBL1 is membrane associated. It's a small protein that in principle should be soluble globally, the globular protein that should not be associated with proteins, but analysis had shown that it binds to membranes by some membrane anchor. The CYPK23, however, is found in the cytosol, and when it associates with CBL, then it can be associated with the C-terminus of the ammonium transporter, phosphorylate there, and switch the ammonium transporter off. Okay, so that's some kind of molecular mechanism that occurs on the post-translational level, on the protein level. So we have phosphorylation that switches off the transporter. Similar phosphorylation by the same kinase opens the potassium channel or changes the affinity of the nitrate transporters.
I have a question.
Yes?
So every time the AT1s are phosphorylated to shut off the ammonium intake, at the same time, they also open for the potassium channels? Is it all
Yeah, that's what we believe.
Okay, it's like this opens and the other one closes?
Yeah,
okay.
That's what we believe. So we have this interaction between nitrate and ammonium. Apparently, we have to balance the charge. And we still need nitrogen, so we have to take up the nitrogen somehow. And we have been for a long time been interested and did a lot of experiments comparing the growth of plants only on ammonium, mixed ammonium nitrate, or only nitrate. And what we normally learn from these growth experiments is that when we have mixed ammonium and nitrate, the plants are typically growing best. They are normally growing pretty well also with nitrate only, but with ammonium they relatively quickly at higher concentrations get problems. And to understand these problems with ammonium, we and others did some transcriptome analysis. And the idea behind and what has been done is summarized here. So many transcriptome analyses, the first ones have looked at nitrogen-regulated genes by an experiment where plants were grown in liquid cultures, in hydroponics, which means the plants are grown in nutrient solutions that are aerated so that the roots get sufficient oxygen, that they do not die, basically. And in these hydroponics systems, you can relatively quickly just take the plants out of a certain nutrient supply, put the plants to another pot that does not contain a nutrient or that contains a nutrient, and you can resupply a nutrient. And what has been initially done was very often that plants have been starved for weeks for some nutrient, and then the nutrient was resupplied to see what happens after this resupply. This is, of course, not really the situation in the field. You never have this on-off of nutrients. But for molecular biologists, it's the only way of really looking directly at effects. And that's why we do that. It's not really mimicking what happens in the field, I would say. But it gives us a lot of ideas what happens first, what comes later, and so on. So, for this resupply of nitrate, which was done first, already 15 years ago or so, we learned that about 1,000 genes are induced or repressed after resupply relatively quickly after 20 minutes. So, changing the nutrient solution of plants, putting plants from one pot to the other and having nitrogen in the other pot increased significantly the expression of 1,000 genes after 20 minutes. And after 2 hours, it was in 1,800 genes or so. So a lot of genes are quickly responding. That's what we look. It's really an effect of the nitrate ion identified by some knockout mutants. It was looked in root and shoot, what happens. And yeah, you can see a lot of genes are regulated. And then it had been analyzed what happens when you resupply nitrate only or compared with resupply of ammonium. These data are not so old, and we added some data to this, and I later show also a little bit of data to these different nitrogen sources. Okay, and what is now important is that many of the homologs of the major regulated genes are also found in crops. Not always directly, the corresponding homolog is then the candidate for the same regulation in crops, but more or less it looks similar in crops, I would say, yeah, like in our businesses. That's good for our model plant, that the model is not so distant from the crops. Okay, so let's go a little bit more into detail. What really happens when we now take away the nitrogen, do not the resupply thing. The resupply is also interesting, but I think agronomically for the farmer, it's interesting to know the first sign, the first gene that this is regulated when you have a deficiency. The first thing what the farmer can see, or maybe what a molecular biologist can detect in the field later also, that would be interesting. The first sign of deficiency. What is the first gene responding? And what happens to nitrogen deficiency had been analyzed for some moderate time scales, days and weeks. And this we know pretty good. When we have nitrogen limitation, and look at the transcriptome and the metabolome, what happens is Well, the genes that are normally involved in making these nitrogenous molecules, they are slowly decreased in expression. The genes functioning in degradation of proteins and amino acids to get some nitrogen from this degradation, these genes are going up. So the plant remobilizes something that it does not need for nitrogen, for new genes. Genes for photosynthesis are going down, typically. Some stress-related compounds are going up, and some signal transduction is going up or going down. It's not so clear here. So photosynthesis is going down, plant growth is going down, nitrogen is recycled, and there are some stress-related substances, for example, anthocyanins giving red coloration. They are produced. So this is pretty well understood, both on the physiological and the transcriptomic level. But what is now really, how quick is this response now? What happens first? What is really the first sign? This is what we wanted to address. And therefore, we did very similar experiments. We grew plants. I think it's on the next slide. Yes. We grew plants in hydroponics, which means aerated nutrient solutions. Not too much nitrogen, not too less nitrogen. 3 millimolar nitrogen was for Arabioxis quite a good concentration. Also typical to soil nitrogen concentration in the soil solution, in crop soil. Yeah, the plants look like this. We have them in certain slits from certain falcon tubes where we grow them. We have small holes in them where the root goes through, and by this we can easily transfer them to a new pot. And that's how they look like when they are grown in 3 millimolar ammonium nitrate. They look very similar when we transfer these plants to only ammonium or only nitrate. And we looked what happens with the ammonium, what happens with the nitrate in the roots of these plants when we transfer them. We looked after 15 minutes. We thought maybe something is as quick as 15 minutes. You see, very small, quick. So these was the nitrate in the roots in the control condition at 0 minutes. After 15 minutes of transfer, the nitrate was the same. It was not used up yet. After 3 hours, yes, there was already a significant decrease. So after 3 hours, we could see, yes, nitrates a little bit went down. So after 3 hours, that might be a good time point where we should see some first gene expression changes for nitrate regulation. For ammonium adapted plants, we had grown these plants first in ammonium nitrate for several weeks, and then for 1 week only on nitrate or only on ammonium. These were the nitrate adapted for 5 weeks only nitrate. These were ammonium adapted 5 weeks only ammonium. So in these plants we still had a little bit of nitrate. It was very low because we had grown these plants for several weeks in ammonium nitrate. They had accumulated nitrate in the vacuoles, and after 5 days of further growth only on ammonium, most of the nitrate was lost, and there was a little bit still, but it was not changing within these 3 hours. When we look at the ammonium, the yellow bars here, we see, well, no change. Ammonium does not go down. The ammonium was in a range where we also have it here in the nitrate adapted plants. In the nitrate adapted plants, we even had more ammonium here. And it also went a little bit down. That was interesting, somehow. Probably it's related to the metabolism, the reduction of nitrate to ammonium. Okay, so, summary of the slide is, for nitrate, we see, after 3 hours, the first indication that we might run into a deficiency, that nitrate is now a little bit going down in the roots. For ammonium, we saw nothing. Probably it's not only the ammonium uptake that regulates the steady state ammonium concentration in the root tissue, it's also the ammonium from degradation of proteins, from degradation of compounds from deamination of metabolites, so that ammonium levels are pretty stable, even if you take away their outside. So, yeah, let's do the transcriptional analysis of these plants here, of plants under control condition, nitrate adapted, and plants ammonium adapted. So the experimental setup is shown here again. So what is the experimental setup? It was about 4 weeks of growth in short day conditions, so that the plants did not flower. Yeah, they nicely grow. In a total of 3 millimolar total N, which means 1.5 millimolar from ammonium nitrogen and 1.5 millimolar nitrate. Both together, in total, 3 millimolar nitrogen. So this was the nitrogen source for 30 years, several times changed, that the plants always have maintained external supply of nitrogen. And then we separated the plants. Half of the plants were grown for 5 days only with ammonium, same amount of N, nitrogen, but now 3 millimolar ammonium, with ammonium sulfate. Or here, half of the plants were put to nitrate only, potassium nitrate. And yeah, then 5 days waiting. And here starts our experiment. Here's the control condition. This is time point 0. And now we compare those plants here with those. The ammonium adapted plants with the nitrate adapted plants. And when we do so, we get, using microarrays, we get gene expression differences between this situation and this situation that can be summarized in such graph here, where we have overrepresented categories of metabolic categories that are overrepresented mostly in the nitrate condition. Overrepresent, blue means overrepresentation, red means underrepresentation. And many categories are somehow overrepresented under nitrate, which indicates somehow, well, the plants feel better in nitrate. With ammonium, they feel a little bit worse. Many things of the metabolism are not so active, basically. And all these categories here contain several genes that are analyzed then together in these kind of categories. In this overrepresentation analysis, we find that the oxidative pentose phosphate pathway is overrepresented under nitrate supply. This has been shown before. That is not surprising. Why? Because the oxidative pentose phosphate pathway gives us carbon skeletons for ammonium assimilation and nitrogen assimilation data, and is involved in this nitrate reduction to ammonium. So that's basically already known. Physiologically, people had shown this much before. Nitrate metabolism, well, was higher under nitrate. That's also not surprising. With nitrate as the resource, as the nutrient, the nitrate degradation, the reduction to nitrate and to ammonium, of course, is only required. In ammonium, you don't need this. You will express it when you don't need it. Amino acid metabolism was more overrepresented with ammonium. That's also not so surprising, because if you have only ammonium, maybe it gets toxic. You want to assimilate the ammonium, then it's not toxic anymore into amino acids. Then some hormones, overrepresented under nitrate, where the plants are developing a little bit better. More hormones, more positive phytohormones, somehow regulating the metabolism and the growth. And we have a little bit of development and transport that is somehow overrepresented here. Okay. So, all of this now was the difference between this situation and that situation at time point 0. And now for us comes a more interesting comparison between this situation and ammonium deprivation, taking away the ammonium. or this situation and taking away the nitrate? Well, we, for comparison, did also some plants that we transferred from ammonium to ammonium, just for comparison. So that the transfer is not activating something that we later have found some expression due to a mechanical stimulus or something. We wanted to see what happens. And indeed, when you do that, you see there is quite some expression of some, there's a mechanical stimulus, obviously, and there's some expression of some mechanical rearrangement also, gene expression. Few genes are expressed there. We did this after 15 minutes and 180 minutes, after 3 hours, because we knew after 3 hours we already see that nitrate goes down in the root. There we can measure it, that it went a little bit down compared to the initial situation. At least at these 3 hours we expect really some transcriptional changes because nitrate was going down. But maybe we can have a look what happens already after these 15 minutes. Yeah, and then we had some plants, more plants to responding to minus nitrogen, either minus ammonium or minus nitrate. So I hope this is clear what we did. Okay, so let's have a look. This is what happens. What is this showing? This is showing gene expression changes as a log-fold change, log-2-fold change. And the color scale, this 1.6 log-2-fold means a threefold fold, a real threefold change of gene expression. So the color shows this 3 log-2-fold change of number 3 means an 8-fold difference of gene expression. Really very high gene expression difference. Yeah. The logarithm in this kind of analysis you often use for some kind of normalization, yeah, which has mathematical some advantages. Okay, and you see, well, what do you see? We have now 4 different situations. And the 4 different situations are these 50 minutes compared to 0, 3 hours compared to 0, or here, 3 hours compared to this situation, and 15 minutes compared to this. This is the differences that we look at. So this is 15 minutes minus a certain nitrate adapted, minus nitrate. And this is what happens in 15 minutes in control conditions after 15 minutes. You see, from the change of the pot, and in these 15 minutes also the daily rhythm, yeah, that the plants are exposed to a daily light rhythm during this day-night cycle, also a lot of genes change. And during these 15 minutes, quite a lot happens already, even if you just don't change the conditions. If you just have ammonium conditions and put the plants to, again, ammonium conditions. Yeah? That's what's changing here. This is a control condition, and this means nitrogen. And you see, after 50 minutes, nitrate-adapted plants, not so much happens. A little bit, it's mostly, blue here, this is an upregulation that mostly happens. After 3 hours, you also have this upregulation of many genes. The 50 minutes of an ammonium-adapted gene, also upregulation of many genes. So the transfer of the plants to a different pot seems to make this upregulation of many genes. Many of these genes that are found there have something to do with calcium, maybe with mechanical stimulation, so they are not interesting to us because they are related to the transfer. The interesting ones seem to be those that go down. These are shown here more in this brown or reddish color. And here are some with nitrate-adapted plants, some genes that strongly are downregulated. A lot after 3 hours, that's the most surprising thing. And with ammonium, nothing is going down here after 50 minutes. and a little bit after 3 hours, not so much. So the plants apparently recognize a lot that nitrate is gone, but they don't recognize transcriptionally so much that ammonium is gone. And this fits somehow to our initial nitrate and ammonium analysis in the roots, which was here. With nitrate, you see a little bit of effect after 3 hours. With ammonium, you see nothing. However, what does not fit, and I think this should be obvious to you, we see a nitrate change within the tissue a little bit after 3 hours. But we see already some change after 15 minutes in the gene expression. It's not much what we see after 15 minutes, but we see something. With this, we have a look in a little bit more detail, and we confirm this by RT-PCR independently. And there are some genes that after 15 minutes or 3 hours are expressed initially and are going quickly down. Now these are the most impressive down-regulated genes within these 3 hours or even after 15 minutes. After this nitrate deprivation, with ammonium deprivation basically nothing happened transcriptionally. What is it? It's sour 6 small auxine regulated number 6. Not so much known about this. A gene number, not annotated as a function, well, lateral boundary domain transcription factor number 37, 38, 39. All three had been described as transcription factors regulating the nitrate resupply response. So that makes sense that we find this. And all these transcription factors are transcriptionally going down. HRS and HHO are two transcription factors that had been, during our study, also identified by others as major regulators of somehow the combined transcriptional response to phosphorus and nitrogen. Also makes sense somehow. And these here also make very much sense. NIA and NIR are here the nitrate reductase number 1 and the nitrate reductase number 1. So nitrate reductase genes are going quickly down when there's outside no nitrate. But apparently inside the cell there is still nitrate. We saw in the roots there's still nitrate available. Somehow the nitrate is apparently externally sensed. So now we can also compare former studies with our study. In former studies we had this resupply of nitrate, and we now did this nitrate deprivation. We took away the nitrate. So how much overlap is there? I mean, with the resupply we would express an upregulation. With the deprivation we would expect a decrease in transcription, the opposite response. But how overlapping are these two responses? I had expected they should match almost perfect. Well, half of the top 50 regulated genes on the nitrate resupply regulated genes, half of them are found in our nitrate deprivation experiment. So it's not exactly the same whether you resupply nitrate and activate gene expression, or deplete nitrate and see degradation of RNAs or decrease of RNAs. Okay, so let's go a little bit further. There was already recently one crucial transcription factor identified, and it was called NLP7. And this transcription factor was suggested to be the regulator of the nitrate response. And we had a look at our data, and unfortunately it was not much transcriptionally regulated, this NLP7 gene. NLP7 stands for nin-like protein 7. Yeah. It was a gene identified to be involved somehow in the nitrogen responses also of legumes, then in Arabidopsis, and then, well, several groups identified it as the major regulator of the nitrate response. And the idea was that this transcription factor was changing its location in the cell. It was basically always there, but when there was nitrate in the cytoplasm, this transcription factor was transferred somehow. It was when nitrate was there, the transcription factor was sticked in the cytoplasm, and when nitrate was not available, it was transferred to the nucleus and activating gene expression. And this transfer of the NLP7 protein was suggested to be, well, the major regulation of these nitrate-regulated genes. Do we see the same response in mutants that don't have this major regulator? Well, more or less we see the same response. Yeah? We use two knockout mutants, NLP7-1 and NLP7-3. Some genes, well, in NLP7 mutants, the gene NLP7 is not expressed. In the corresponding wild type to this, the gene is expressed. Okay. So, what happens to other genes, to this gene, SOR6, transcription factors, or the nitrate reductase, the nitrate reductase? What happens is that this decay of the RNA is still there. So apparently this decay of RNA is not regulated by this major transcription factor. When we worked with these transcripts, we also looked at other papers, what researchers had found previously and looked at previously, and there we had a surprising finding. And one surprising finding was that, for example, the messenger RNA for this transcription factor had been identified in a previous study as relatively stable transcript. So people had measured the stability of all transcripts by microarrays, and some transcripts are really degraded within an hour, within 2 hours or so, and some transcripts are stable for 9-10 hours or so. Now this was very stable, and this, for example, was also pretty stable. But why do these transcripts now decrease so quickly when we take away nitrate? Is there maybe some active degradation of transcripts where we take away nitrate? And this we tested with blocking pre-treatment or blocking new messenger RNA synthesis with antimycin D. And in these experiments, we added this blocker of new transcription, and we can simply then, because we don't get new transcription, we can simply see the decay of the transcripts. And when we do so, in the presence of nitrate or in the absence of nitrate, we see in the presence of nitrate, the transcripts are more stable, half-life is longer, and in the absence of nitrate, they're degraded faster. Somehow, we believe there's an active degradation of certain messenger RNAs when there's a nitrate deprivation. Oops. So, and with that, we were already quite happy to have this transcriptional regulation, but when you look or want to really understand how the metabolism works and adapts to low ammonium or low nitrate, you also have to look also on the metabolome or on the proteome. Why on the proteome? Because, well, transcripts may change, but the proteins may be very stable, and they don't change at all. So we have to look at the proteome and see whether the proteins are also stable or whether they are quickly degraded when we have outside no ammonium or no nitrate. And so what we did, we asked our colleague, Schulze, in Hohenheim. She is expert in proteomics, especially phosphoproteomics, and we analyzed together with her the proteome of our roots at the same time points, and we try to correlate the gene expression with the protein amounts that we find. This is shown now here in these diagrams for nitrate and for ammonium, and here it is shown which proteins did we find. So from the gene expression analysis, we knew that Arabidopsis had about 22,000 expressed genes, and in the root of these, about 10,000 are significantly expressed. Significantly expressed means we can find the transcripts, but maybe, well, the transcripts are relatively low, that not much protein is made. When we look at the proteins, we can see about 4,800 proteins were always found in our different conditions. So half of the proteins that we were suggested by the transcripts were seen, half of the proteins were not found. So the proteome gives only part of the total proteome, yeah, in this kind of analysis. It's a very, very solid part of proteome that we can look at, but we don't find all proteins. Yeah, that has some chemical problems that we don't find all proteins. Because transcripts are more similar to each other. Proteins can be negatively charged, protein can be positively charged, can be hydrophobic, and so the chemical properties of proteins are a little bit more complicated to detect. Okay, anyhow, we have these nitrate adapted, ammonium adapted. Most proteins are then always found. Some are only found in some certain conditions or in two of the conditions. However, what we now have a look at the log fold changes for protein and the log fold changes for the transcripts. For nitrate, what I told you before is that after 15 minutes you see some transcripts, very few transcripts that will go down. After 3 hours, quite a lot of transcripts go down. That's the transcripts that go down. When you look for these transcripts, how much of protein of these was changing, you see, well, it does not very much correlate. What I expected initially, and what is never found in this kind of analysis, is that when you have change in the transcript, so the transcript goes up, you would expect that after a while, the protein also goes up. When the transcript goes down, the protein should also go down. Well, transcript goes down, protein should go down. So what we expected was more or less, at least after 3 hours, maybe not at 15 minutes, that may be too fast, such a line. We did not at all find such a line. Not for nitrate, not for ammonium. And what was interesting was that for ammonium we had basically no transcriptional change at all. For ammonium we found a lot of changes in the proteome already after 15 minutes. Yeah, so ammonium adapted, nitrate adapted. Especially the quick things that may happen may only be phosphorylation of proteins. That we can imagine can happen quickly, not the complete degradation of a protein that may, or new synthesis of a protein that may need a little bit longer time, but 15 minutes are definitely sufficient to phosphorylate a protein, to dephosphorylate a protein. So what did we find for phosphoproteomics, for the phosphopeptides? We found several phosphopeptides. In total it was almost 600 ones. Some specific to nitrate, some specific to ammonium, some specifically responding to our time course. We could compare this with a previous study of Weithout, where she did a resupply experiment and where she looked at the phosphorylation after resupply of ammonium and nitrate. And she had found these 400 or 600 proteins that were now new or less phosphorylated. In this study, we had a little bit less, and the overlap was relatively small, but there was some overlap at least. Although the technique was exactly the same, because what they did in the same lab, and everything was the same. What was apparent And interesting to us was that the phosphorylation of many proteins that we knew already had to do something with transport. Transport of ions, transport of water, transport of protons, and that were known to be in transporters that have to do something with the fact when we take away ammonium or when we take away nitrate, then in these transporters something might happen. For example, PIP, water channels, were changed in phosphorylation of certain glycoprotein, membrane proteins that have unknown substrates, I would say. Again, a water channel was differentially phosphorylated, ammonium transporters were differentially, nitrate transporters of the nitrate transporter 2 family were differentially regulated, proton pumps were differentially regulated, involved in the setting the pH. Again, proton pump, ammonium transporter is differentially regulated, so that in the end we came up with, wow, this final cartoon. What happens when we take away nitrate or when we take away ammonium? Now I press again. And something should happen now. So in summary, to sum up all these tables and numbers that I showed in some diagram that shows the cell and its cellular metabolism. Oh, it's gone. No problem. So, that is. And the upper part shows what happens when we take away ammonium after 15 minutes, after 3 hours. And what happens is that we have no transcriptional differences. Transcription does not much change, not much genes activated, not much genes going down. But on the protein level, and at the phosphorylation of protein, there happens something. Especially in some membrane transporters. Some membrane transporters, probably for cations, for ammonium, for water, for protons. Yeah? And it's some proteins are phosphorylated. Ammonium transporters are surprisingly switched off. They're phosphorylated, and we know this switches off these transporters. And some other proteins are dephosphorylated, very often activating them. So apparently, when we take away ammonium, the plant has to adjust its cation, anion uptake in a way that processes at the plasma membrane are adjusted by adjusting their activities of these transporters. When we take away nitrate, much more happens. And much what happens, apparently, is in the nucleus, or outside of the nucleus, where RNAs are produced and degraded. The nitrate deprivation was not leading to different nitrate concentration in the cytoplasm yet, after 15 minutes. But apparently, the nitrate outside was sensed and giving a signal. And the candidate for this kind of signals has been identified by others, and this signaling is associated with the nitrate transporter NRT 1.1. And when this is phosphorylated, it has a different transport activity. Maybe some other nitrate sensors are also phosphorylated. We don't know exactly which ones these are. Apparently, when we take away outside the nitrate, there is a signal flowing in. It's not nitrate itself that is then going down here so quickly. It's a signal that tells us, or tells the cell, outside there's no nitrate anymore. Already adjust your gene expression in a way that certain genes are differentially expressed. This transcription factor that normally activates these genes is not so much involved in degradation of messenger RNAs, but messenger RNAs, especially of certain ones, are degraded. Yeah? When this signal is missing, the messenger RNA is degraded. And when there is nitrate inside, and probably this nitrate transporter signals, oh, there's outside nitrate. This signal normally tells no mRNA decay, please produce further proteins. Okay. So, that was relatively complicated molecular analysis of this ammonium and nitrate dehydration and the molecular processes involved. So, this was quite complicated, and now we have, of course, to ask why we do that and what it helps us for the plant and the crops. Now I do it slide per slide. We first have a look what you can take as take-home messages from this kind of study. I have 5 points that I want to mention here. What is it? Major transcriptional repression. So many transcripts rapidly go down when you take away nitrate, but not when you take away ammonium. This is independent of this NLP7 transcription factor that is involved in the nitrate resupply response. And this happens before the tissue nitrate drops. So apparently the nitrate is recognized outside and gives a signal into the cell to do something. There's a phosphoproteomic change, so phosphorylation is not more or less. It's in some proteins it's more, and in some other proteins it's less. But there are specific sites phosphorylated or dephosphorylated, especially of ion or sodium transporters, in ammonium deficiency or ammonium deprivation. These are probably related to the ionic changes that occur when you take away the ammonium. This nitrate depletion reduces this messenger RNA decay of some, normally under normal conditions, under nitrate supply conditions, stable transcripts. So only under no nitrate supply, these transcripts are rapidly decayed. The depletion is not exactly the same or the opposite of the resupply response. And what I think is quite important is when we now compare this nitrate and ammonium effects, is that in the literature we should, in many cases, better talk of specifically nitrate depletion, where we mean nitrate depletion, or specifically talk about nitrogen depletion when we deplete of both, because we see that different nitrogen forms are having completely different responses, ammonium and nitrate. And nitrogen depletion is not exactly the same as nitrate depletion. So nitrate has some specific effects. Okay. So one word, somehow, I think I will switch this off. That's why it doesn't work. How did I get it to work again? Okay. So, do these kind of studies help us that we can improve crops by identifying certain specific nitrogen regulated genes? And there we first have also to do a literature review. What is important? Well, the first thing is just a physiological statement that nitrogen can be taken up as ammonium and nitrate, and when it's as nitrate, it has to be reduced to ammonium, and this is a major energetic cost. Okay. So we have a lot of genes involved in these nitrogen responses. We know a lot of genes directly involved in these conversions of nitrate to nitrate and ammonium. We know all these genes. Further ammonium assimilation and so on. The uptake transporters, all these are identified. So can we by genetically modifying them, improve nitrogen efficiency in the plants also? In reality, under some laboratory conditions, we sometimes can. However, when such kind of plants are tested in the field, there's no example, no plant, no transgenic plant, in which a single nitrogen metabolism-related gene has been changed, that these plants are really better than the best varieties selected by the breeders. This has many reasons, yeah, why this is so, but it suggests that currently, by taking single genes, changing their expression in transgenic plants, and expressing these transgenic plants and trying to improve nitrogen use efficiency with that, with physiological knowledge about these genes, is not very successful in field conditions. There are, however, basically two examples, some of these supported by several publications, some only by yet one publication, that there can be genes that may improve nitrogen use efficiency, and there are some examples from rice, mostly from Chinese scientists, where in rice, which is normally a species that is better adapted to ammonium-containing conditions, or high ammonium conditions, in flooded fields, for example, there are little oxygen in these floods, and not much oxidized nitrogen, you have a lot of reduced nitrogen as ammonia. And rice is adapted to very often these conditions, preferentially takes up ammonium, and in rice varieties, they have shown several times when you then express certain nitrate transporters in rice, you can get better nitrogen use efficiency under field conditions. Whether this is really with the best varieties that farmers already use is not really always clear. And one recent publication from last year was really a surprise for many people, because there was a Chinese group that suggested that certain wheat and rice varieties which have been shortened during the Green Revolution, so that the varieties are shorter in their stem, so that they can hold more grain mass and don't lodge. Yeah? And these Green Revolution varieties are typically mutants of the gibberellin signaling pathway. There are also some other mutants that seem to not have to do with gibberellin, but typically stem elongation, internode elongation is very sensitive to gibberellin. It's supported by gibberellin. And in these gibberellin mutants, you have shorter stems, you have shorter internode fractions, and you have less lodging, and you have varieties that can produce more grains with certain nitrogen fertilization. And by manipulating some gibberellin-related genes and a certain transcription factor, they could improve nitrogen use efficiency in their field and field conditions, both in rice and wheat, at least in one condition that they published. Yeah? This seems to be really surprising. Or this is very surprising, and well, I will show you why this is so surprising, and maybe then we should make another break. Or we should make first the break. Well, I may repeat it later. Because in wheat breeding, the target, since a long time, is nitrogen use efficiency. Why? Wheat grains are typically selected for high nitrogen content, for high nitrogen concentrations in grain. And why do we need, or why do we want high concentrations of nitrogen and protein in grains? Because we normally take wheat for flour production, and this flour is used for baking. And baking requires, or bakeries normally require wheat varieties that have more than 12% of protein. And more than 12% of protein is around something like 2-point-something% of nitrogen in the grain. And more nitrogen, more protein in grain, you typically get if you fertilize a little bit more nitrogen. That's actually what the farmers do, especially what the farmers in Germany do, because they also paid for the protein quality or the grain quality in the wheat grain. And the more protein is inside, the more they get for their grain. So they fertilize them. And that's one of the nitrogen inefficiency problems in Germany with wheat, because wheat is one of the major cereals and crops in Germany. And the most important thing that I wanted to mention is that for a long time, breeders have tried to increase grain protein genetically. And farmers and agronomists had to try to increase grain protein. But the problem is basically always that grain protein negatively correlated with yield. Basically always. This is a physiological law. And this seems to be a little broken by the recent Nature publications that I mentioned in rice and wheat. And we'll see how this develops. I would suggest we make a short break and then come back in 15 minutes and have a look again at this. Thanks. Our question was why nitrogen use efficiency is so low, especially in wheat. And I already mentioned a few things. One is that then we have this very strong correlation, a very strong, extremely negative correlation of grain protein with yield. And this negative correlation was, for decades of breeding, not possible to break. And what you see here on the left, a diagram with grain protein nitrogen. So this here is something like 18% or 16% protein, really high amount of protein in these grains. And we need, for baking quality, typically something like, now it should be okay. Is it okay? No. Now the microphone. Sorry, thanks. So this negative correlation was not possible to break. What you see here is different genotypes and different colors, fertilized at 250 kilograms per hectare, or 200 kilograms per hectare, or 100 kilograms per hectare in a breeding program in Britain, in England. And, well, with less fertilization and less nitrogen, you get less protein in the grains and less nitrogen. Nitrogen, protein, directly correlated. And you don't get enough baking quality. You don't get these 12%. With 200 kilograms per hectare, you get them in some genotypes and others not. And this was always, for a long time, one of the drivers why the farmers used quite high amounts of nitrogen fertilizer to get above a threshold of 12% of protein in the grains, to sell the grains a little bit more expensive to the mills. And then the baking quality was thought to be better. However, since more than 10 years, we have data, not only in Germany, but especially also in Germany, that many different varieties, like here, having protein concentrations from 12% around and up to 14%, basically, these are not very well correlated with baking quality. So if it would be correlated, this grain protein concentration would be correlated with baking volume, then we would have here all these genotypes on a line. They are not. Basically, you can get very good baking quality with Mirage genotype at low grain protein, or relatively good low grain protein. And you can get much less baking quality with Tommy with much higher protein. So apparently, the system, how it works in Germany, that the farmers are paid for protein content or protein concentrations in the grain, is a little bit misleading because it does not really show the baking quality. Many modern varieties have very good baking quality. And somehow, probably we get better nitrogen use efficiency. We suggest that the farmers should not be paid according to grain nitrogen. And especially, we don't need such high grain protein for feeding animals with this wheat. So for certain other wheat that is grown in the field as feed, we don't need this baking quality as a parameter. So in Germany, we have this system that we have split applications of nitrogen, as I explained a little bit earlier, up to 60 kilograms of nitrogen at the very beginning of the growth phase in early spring, then again 60 kilograms at canopy closure, and a final, quality fertilization to increase protein, sometimes as a foliar spray with urea to the plants to increase kernel weight, grain protein concentration. ripening. And many experiments have looked at this last fertilization step, and nowadays it's very often found that it does not increase grain protein. So farmers should omit it. These 40 kilograms are only for the environment. Okay, so the last thing is that, of course, for baking quality, the gluten is very important. This gluten is a fraction of the proteins that are found between, let's say, 8 and 18%. 18% is really high protein concentrations in the grain. Typically, the grain is also relatively rich in minerals, in lipids. There's some moisture, about 15%, and of course, it's a source of starch. Some fractions of the proteins are not associated with baking quality, but the glutens, of which we can fractionate more in the gliadins and glutenins, and we have several of these, and many genes encoding individual omega gliadins or alpha gliadins or so. But by manipulating them, maybe with modern gene editing tools, we can knock out certain protein fractions that are not important for baking quality, and we can modify maybe the different fractions of glutens in the grain, that we get higher baking quality, even with much less protein content and concentrations in the grain. So that's one thing that we envision for the future. So, last but not least, I want to come again back to some experiments. I know that it was pretty tough, the molecular analysis before with Arabidopsis, but now we do some kind of analysis again, comparing ammonium and nitrate as nitrogen sources for Arabidopsis. This time not in hydroponics, in nutrient solutions, but this time we take agar plates and we grow small plants from the beginning on, from the seed germination on agar plates, and these agar plates only contain nitrate, contain ammonium nitrate, ammonium plus sugar, or ammonium without sugar. In all cases, we have here for this picture 6 millimolar total nitrogen, and this you can see is already a little bit toxic when you supply the nitrogen as ammonium. The plants do not want to grow so nicely as they do here with nitrate. And we quantified the primary root lengths for these kind of plants. And we see with nitrate they were longest, with ammonium nitrate they were a little bit shorter, and then at higher concentrations of ammonium nitrate the primary root decreased. Yeah, and with ammonium, either with sugar or without sugar, the root growth was severely inhibited. And we measured that these plants here took up a lot of ammonium. Ammonium concentration in these plants was pretty high. So apparently the ammonium at some concentration was toxic, and we thought, well, this is here half maximal effect at around 6 millimolar or around 3 millimolar. That's here a range where we should look a little bit more in detail what happens to these plants. And what we did was, we again used microarrays to analyze gene expression of these plants. Here we did not only focus on the root, we took whole plants because these plants were so small, seedlings. And with microarrays what we found was partially overlapping with our other analysis that certain metabolic functions are different in ammonium or nitrate, or both ammonium nitrate supplied plants. Some differences were also in the cell wall, which we found interesting, but we don't really, or we have not really addressed what happens there. One interesting thing was that there were already some responses apparently different in the hormone signaling, especially the auxin signaling. And this sounded to us somehow interesting, and we have a look further at what happens there. When we look in the roots, we can stain roots of Arabidopsis, certain transgenic plants of Arabidopsis, which have a certain auxin-sensitive promoter fused to a reporter gene, and then we can get a blue color when this reporter is active. And this reporter plants are called DR5 for this auxin-responsive element, gas plants. And these plants were used here to see whether there is some auxin-sensitive staining in ammonium nitrate or ammonium. And we found, well, there was a little bit of differences here, maybe a little bit less of the staining in ammonium sometimes, which may reflect a little bit less numbers of laterals in ammonium. When we look at the primary root, we see with this DR5 gas reporter, we see this auxin response, and this was depending on the synthetic auxin at 0 concentrations or slightly higher concentrations. We see, well, apparently auxin peak, typically found in the root, is getting bigger the more auxin, synthetic auxin we supply here. Okay. With ammonium nitrate, and when you compare these pictures, maybe you recognize that with ammonium nitrate, the auxin peak here always is much stronger than here, and also here, stronger than here. So we did it at 2 pHs because sometimes auxin effects are thought to be very important, at very acidic pH, where root growth is inhibited by the too acidic pH induced by the auxin. But this occurred also at alkaline, slightly alkaline pH, but also at acidic pH. So apparently, in a manner dependent on the synthetic auxin and depending on the ammonium or ammonium nitrate that we supply, the auxin response in the root tip is different. When we look at these different synthetic auxin concentrations at lower pH and higher pH, we see that the lateral roots are less with little auxin, with more auxin. We get more lateral roots. So lateral roots require the auxin to be made, and this dependence on the auxin is steeper with ammonium somehow than in ammonium nitrate conditions. And when we now use auxin mutants that have normally less auxin in the root tip, or auxin mutants that have more auxin in the root tip, genetically, we can restore the normal lateral root formation to these mutant plants. Because ammonium decreases the auxin peak in the root tip and overall the transport of the auxin from the shoots to the root. In mutants that have genetically more auxin, and with ammonium we have less auxin, we could balance maybe, like in wild type, back, come to the lateral root number as in ammonium nitrate conditions. And exactly this is found for these mutants that have more auxin in the root tip genetically. When we put them on ammonium, ammonium reduces the auxin transport from the shoot to the root. We get the same amount of laterals as wild type under ammonium nitrate conditions. And overall lateral root amounts is decreased in mutants where less auxin anyhow is there. Okay. So, from this, some responses under ammonium stress in these kind of plate assays are that the reduced auxin transport from the shoots inhibits root growth and branching with ammonium as salt nitrogen form under these conditions. In soil, it's extracted different, and you can't in soil really separate the ammonium from nitrate completely, typically. Mostly you have both substances there. This ammonium suppression can be overcome by adding external auxin or genetically increasing auxin accumulation in the root tips. And part of this reduced root growth with moderate ammonium is mediated by the phytohormone auxin, or the lack of the phytohormone auxin. Good. So this is what I wanted to tell you for today. And I'm again back to my acknowledgement slide, and I thank you for your attention.